Hi, everyone at home. For those of you just joining, welcome to LUMCON Science Talk. If you want to interface with somebody live here at BlueWorks, please use the question box you see in your dashboard. If you can, want to practice using that question box, go ahead and type something in that box just to tell us, hey, we have a great online audience as well as an in-person audience. Thank you, Michael. It's great to see you. Hi, Grayson. Hope you're doing well tonight. Thanks for attending. We're just going to wait a couple more minutes. We have quite a few people logging in yet. Looks like we have people from all over the country attending tonight. That is great. Sorry, you guys, messing with the camera. Hi, Diane. Thanks for attending tonight. Beth, thank you for attending. All right, looks like we have login kind of slowing down, so we're gonna go ahead and get started. All yours.
Oh my, very cool. Thank you so much, people in the real world and people in the virtual world. My name is Brianne Duclos. I am the collections manager here at LUMCON, and I am very excited to kick off a new season of LUMCON Science Talks here at our BlueWorks facility at our Houma Maritime campus. The title of the talk tonight is LUMCON's Natural History Collection, Then and Now. There are 4 components in that title, and I hope by the end of the talk, we'll all come away with a little better understanding of each of them: natural history, collections, then, and now. We'll start by talking about natural history. Natural history is a very old scientific discipline. We're talking thousands of years old. Ancient Egyptians practiced natural history, and it's a pretty straightforward kind of science. Natural history is the observations of organisms in their natural surroundings. Anybody can do natural history anywhere. If you see an animal or a plant or a mushroom anywhere and it's doing something, even if it's not doing something, you're doing natural history. And that information is really important for all kinds of modern day biological and ecological science. When you do natural history, it's important to record a set of information associated with your observation so that it has other uses with these modern day scientists. That information includes where you observed your organism, the kind of habitat you observed that organism in, its name, your name, and the date at which you made your observation. Creating these natural history observations pretty quickly and organically evolved into actually collecting that organism itself. And this is how natural history collections came to be. Natural history collections can also be referred to as scientific collections or biological collections. We call them natural history collections here at LUMCON. And collections really brought the natural world indoors. If you didn't want to go outside and make observations about what was going around you, you could go to a collection, right, and interact with those specimens that other people had brought in themselves. They're really sources of wonder and curiosity. Institutions built up around these collections, right? Historically, there have been a lot of private collections, but museums also came into existence to showcase the wonders of natural history collections to the public, and still largely do that today. Academic institutions like us here at LUMCON have a pretty close association with collections historically as well, with universities housing natural history collections, and people that work in collections, curating them and managing them, often have academic training in the taxonomy or the biology of the organisms within that collection. Additionally, many leading collections management programs and collections management technology currently is developed at academic institutions. There are a couple of types of natural history collections that I want to talk about tonight. The first is dry collections, and this is a lot of natural history collections. These include specimens that can be directly handled. You can actually touch the specimen. These include things like fossils, plants, bird skins, insects on pins. Fungi are often stored this way, mammal skins, and also dry shells. There's also wet collections, and these are specimens that are preserved in liquid, preservative in jars. We have pretty extensive wet collections here at LUMCON with our collections of fishes and our collections of marine invertebrates. Reptiles and amphibians, or the discipline of herpetology, are also commonly stored as wet collections. What kind of things can you do with collections? We'll talk a lot more about this later in the presentation, but I do want to make the case early on for why these resources are important. Collections preserve a record of the past, right? Especially if you record information with your observation or collection, like when and where it was collected. This information has many modern uses. You can use this information to assess species populations and species communities, communities of different species together, how those look and how those might change over time. You can support taxonomy, the science of classifying these organisms, which is always changing. A lot of that work is supported by specimens in natural history collections. Finally, I want to highlight the field of biodiversity informatics. This is where my background is related to collections. I work a lot with the data associated with these specimens and do really cool stuff with it. Lastly, I want to talk tonight about how collections make really excellent teaching tools. And in particular, the collections that we have here at LUMCON are a really cool example of demonstrating and showcasing the complexity of animal biodiversity on the planet, much of which is found in marine waters. And we have examples collected locally of so many of these different groups of animals. You might remember from your high school biology class, the taxonomic levels listed here: kingdom, phylum, class, order, family, genus, species. Tonight, I'm going to highlight all the different phyla of animals that we have in our wet collections. That's pretty high up on the taxonomic levels there, but it gets, there's billions of species on the planet, right? And there's about, let's see, move to my next slide here, and I'll just show you. This is a list of the 14 animal phyla that we have in the kingdom Animalia, which sometimes is referred to as the kingdom Metazoa. The stuff in between is how you get between kingdom and phylum. Don't really worry about that. That stuff tends to get pretty complicated. But what I want to use this slide to illustrate is to tell you that we have examples in our collections here at LUMCON of 13 of these 14 animal phyla. The only one we don't have are the rotifers, and rotifers are primarily freshwater species. So while we have some freshwater animals in our collections, most of the diversity listed in those animal phyla have marine habitats, and we have examples of many of them here. We have 4 collections within our natural history collections, and I'll talk about each of these in turn. We have botany, our plants. We have invertebrate animals, we have vertebrate animals, and we have a new collection that's growing called the Oyster Reef Biodiversity Collection. I want to mention a recent heroic effort to restore and relocate our collections. For many years, our collections were located down at the DeFelice Marine Center down in Cocodrie, and as of this winter, they were relocated all up here to the Blue Works facility at the Homa Maritime campus. We had a couple of professors that received a grant to support this work. We had an amazing technician who worked with us for a couple of years who did all of this work. She had a huge task. We have over 1,500 jars of specimens in this collection, and her objective was to assess each of these jars, determine its condition. Many of them were not in the greatest shape, and she had to recurate these jars. That is, give them each a fresh, brand new jar and replenish and refresh the preservative liquid in these jars. We use 70% ethanol to keep our specimens preserved. She also, once that was completed, she relocated everything, and she got a catalog system going now. So every single one of our specimens in our collection has a unique catalog number that we can use when working with the specimens themselves and the data associated with them. I'm going to go through each of our collections, and I'm going to start with our little botany collection. Unfortunately, this collection is a sliver of what it once was, but we still do have these 50 specimens. From 40 species in 30 genera in 20 families. I've done a lot with numbers and species and records in my day, but I've never had a really nice, like, even pattern of numbers here. And when I ran those, I was really pleased with how even and symmetric that looks. That's a kind of a neat pattern. Most of these specimens were collected locally in Lafourche, Terrebonne, and Vermilion parishes. Most of the specimens were collected specifically in the year 1981. By Dr. Alex Lassen, who by all accounts was an absolute botanical legend in southern Louisiana, and in particular southeastern Louisiana. Dr. Lassen graduated from Golden Meadow High School. He went to Nichols State for his bachelor's. He went away for graduate school and then came back and was a professor of botany for 3 decades at Nichols, during which time he expanded the size of the Nichols herbarium from just a few specimens to almost 25,000. Absolutely Herculean effort. And he contributed a lot to the botanical knowledge of our local area. So I feel really lucky that we have some of his specimens in-house. I want to highlight a couple interesting stories of our botanical specimens. This one on the right is the marsh elder. It's a pretty common shrub in the wetlands down around Cocodrie. The specimen on the right is actually noted on the label as being a viney parasite of the marsh elder, and that's how Dr. Lassen pulled it off of the shrub that he collected it from. I thought that told a really neat story. Most of our wetland plants are going to be grass. Our specimens are pretty grass dominant. This is the genus Cyperus, and I just chose these 3 specimens to highlight some of the diversity that you can see within a genus of grasses. And of course, if I'm going to mention grasses, I'd be remiss not to mention Spartina, the big cordgrass. These are 2 examples of Spartina species, and we have quite a few specimens of the big cordgrass in our botanical holdings. Next, I'll tell you about our wet collections. This is the bulk of what we have in our collections facility, and includes both our invert and vertebrate zoology collections. These specimens were collected between the years of 1966 and 2010, with peaks there in the late 80s and in the late 90s. These specimens were collected really locally. This is a word cloud of the descriptive locations associated with each of the specimen records. And you see a lot of familiar names, right? Terrebonne Bay, Cat Island Pass, Ile Dernier, the Gulf of Mexico, Cocodrie. This is a really, really local collection, and I think that's a really unique thing to have. The specimens were largely collected by students and their professors through a rich tradition of coursework in marine zoology and marine invertebrate zoology. And you can see all the different ways you can write marine invertebrate zoology there. And I can tell you all of that information because a lot of the records that we have were really detailed. The physical labels associated with many of these specimens include really important information, like the name of the organism, the names of the collectors, when it was collected, where it was collected. And I also inherited a really rich and robust digital record, which in the case of older collections that may not have been managed for a while is not generally the case. So I feel really fortunate that I was able to start here with that. Let's talk about our invertebrate zoology collection. This is where a lot of our animal diversity is, right? All 13 of the animal phyla I mentioned in that phylum slide are represented in our invertebrate zoology collection, ranging from sponges and jellyfish up at the top, down to urchins and early chordates at the bottom. Chordata is a really interesting phylum. It's often associated with vertebrates, things that have backbones, right? Chordata actually describes the presence of a nerve cord. So there are some chordates, just a few, that are actually considered invertebrates, and we have a few of those in our collection. We have 722 jars or specimen lots in this collection, representing 358 species. I'm going to highlight some of the interesting specimens in this collection. This slide highlights some early diversity. We have a red-beard sponge from the phylum Porifera in the middle there. That specimen was collected in 2023 by one of our current graduate students. We have an onion anemone from the phylum Cnidaria. That phylum includes corals, anemones, and jellies. We also have a comb jelly on the far right there. That's from the phylum Ctenophora, which includes just comb jellies. There's an interesting debate in the taxonomic literature right now as to whether sponges or ctenophores are the first animal to evolve. It's pretty controversial because we've always been told that it's sponges, right? And it'll be interesting to see how that unfolds. I also included a couple examples of worms here. Worms came later evolutionarily, but we have a ton of worms in our collection. They don't photograph well in the jars, so they're really tiny, and most of the jars we have of them are quite small. This is a ribbon worm from the phylum Nemertea. And these here, you can barely see them. There's about 10 to 15 of them on the bottom of this tiny jar. These are airworms from the phylum Chetonapha, and this is the only specimen we have from that phylum in our collection. I want to highlight a couple of squid. I really like the squids in the jars. I think they're really cute. But they're also really interesting species, right? We have a longfin inshore squid there on the left, and we have our Atlantic brief squid there on the right. The brief squid is one of our uncatalogued specimens, but it does say on the label that it was collected off Wine Island Pass in 2009. And the longfin squid were collected by a marine zoology course in a trawl in 1986. This is a long spine swimming crab, one of the many specimens we have in the phylum Arthropoda. Within this invert collection, by far, most of our holdings are arthropods. Arthropods include barnacles, they include crabs, they include shrimps. Shrimps and crabs are very popular and common and important species around here, so that's why we have so many of them. This specimen in particular was collected in 1988. During a marine invertebrate zoology course, and they collected it at a depth of nearly 300 feet under the water. Lastly, with our inverts, I want to highlight some of our echinoderms, the urchins. On the right, we have a couple of jars of red heart urchins, so named because when they're living, that outer shell is covered in teeny tiny bright red spines. They're really beautiful. These were collected pretty deep. The range of the trawls that collected each of these specimens goes from 425 to 820 feet. So pretty far under the water, we can do some pretty neat deep sea work with our Pelican research vessel. The burrowing brittle stars there on the right were collected 40 feet deep off Cat Island Pass, also using trawls during marine zoology courses in the late 1980s. Let's talk now about our vertebrate zoology collection. Now, when I started at LUMCON, I'm pretty new. I started at LUMCON in the spring, in the month of March. I just assumed that this was an ichthyology collection, a collection of fishes. Turns out it's not. We have 3 records of our 1,100 records that are of non-fish invertebrates. 2 of them are reptiles. We do not have possession of those anymore, but they were once in our halls. We have 1 jar of an amphibian. Spoiler alert, I'm about to show it to you because it's really cute and really interesting. But then we had to think about all these fish. And oops, let's go back here. I want to talk a little bit about the taxonomy of fishes because it is pretty wild. It's pretty crazy from top to bottom. Depending on how you look at it, where you go for your authoritative resource on fish taxonomy, there's either 4 classes of fishes or 2 classes of fishes. And these are all determined by whether or not they have cartilage or bones, and then also by the types of bones that they have or the structure of their cartilage. Let's talk about our amphibian specimen in our collection. This is the lesser siren, Siren intermedia. This specimen was collected in 2006 in the Atchafalaya River Delta in St. Mary's Parish. One reason of many that the siren stands out from the rest of our invertebrate holdings is that as an amphibian, it is a primarily freshwater species. It does have some salt tolerance though, so it's likely that this was collected in some brackish waters, some slightly salty waters. This specimen brought me a little bit of a challenge, and I included here to illustrate how the taxonomy of fishes is complicated and always changing. This is another one of our uncatalogued specimens, and all the information that I had on the label for this specimen was a name, Stenatomus CF aculeatus. And what that CF tells me is that the identifier is not certain that that's actually what this fish is. So I looked it up, a little Googling, right? We all do it. And turns out that this species name is really obscure. It only comes from a few references from the late 70s and early 80s, and it was never actually considered a verifiable species of fish. So I looked, a little bit more into the genus here, and what I found, and what I was able to corroborate thanks to our collection, is that this is more likely an example of a Gulf scup, Stenatomus caprinus. We do have some verified lots of this particular species in our collection. And the Gulf scup is pretty common in and around the Gulf of Mexico. Next, we have the inshore lizardfish. We have 10 lots of this species in our collection. It's fairly common, and I chose to highlight this species to show how for some of our specimens, we have lots of different sizes of these fish. So you can see the different size classes that these fish can have. The lizardfish can actually get quite long. But that jar on the right is a 1 gallon jar, and that's the biggest jar that we can store something in. So we will eventually have the potential to store larger specimens, but for now, it has to fit in a 1 gallon jar like those fish on the right there. Lastly, I want to highlight a couple of specimens from the less common and specious classes. So the one on the left here is a long-nosed gar. We just have one gar in our collection, and gar are another freshwater species. So that explains why we don't have so many of them. We have 4 specimens of this Gulf chimera. Chimera are really interesting fish. They're an ancient lineage of fish that are still hanging in there. And let's see, I don't have any information on the depth at which these were collected from their labels, but I know that they were all collected on pelican cruises for marine zoology courses. Lastly, within our collections, I want to highlight the Oyster Reef Biodiversity Collection. This is an ongoing collection. It started in 2022 by Dr. Stephanie Archer and the scientists in her lab. What they do is they go out to oyster reefs and they place habitat trays on these reefs. They leave them out there for 2 weeks, return and collect all of the organisms that have gathered in those trays. They bring them back to the lab and they process and identify everything they find in those trays. And when they find something new, they keep it, they preserve it, and that's called a voucher specimen. This is a collection of voucher specimens that the Archer Lab uses to corroborate and confirm identifications of the species in their lab for these biodiversity surveys. I last checked in on the Archer Lab collection in June of this year, but I know that they worked really hard this summer, so these numbers are probably an underestimate. I have 84 records of different species that have been collected in these surveys, and there's quite a bit of biodiversity there. We have a lot of fishes, but we also have a lot of arthropods, a lot of gastropods, and bivalves. A lot of diversity in this tiny little collection. So now that I've introduced you to the collections that we have here at LUMCON, I want to talk about some of the things that we're doing with collections today. And one big one is digitization. I've talked about how, when you record collection information, right, you write it on a label, you write it on a data sheet, you write a physical copy of that information down somewhere. The way that most science is done these days, right, if you're analyzing data, it's done on a computer with digital files. And so a lot of collections have been working really hard over the last 25 years to get their holdings digitized. I mean, when you think about the number of holdings that some of these collections have, right, this is a huge backlog for some of the largest collections globally. But it's really important so that people can get this data and use this data. There's a number of online resources that have been created called repositories, where you can put data associated with collections and then download that for use. One of them is GBIF, the Global Biodiversity Information Facility. Another is OBIS, the Ocean Biodiversity Information System. There are a set of standards now used to tell people how to format their data. So if we all follow these data standards, people here that are using and sharing data on the internet, somebody on the other side of the world can download that data and know exactly what information is being shared in each of these pieces of data. Something that I think is really cool about digitizing data is this concept of a digital extended specimen. Specimens are becoming now so much more than their physical selves. You can have the physical specimen itself, you can take an image and a digital record of it, and then you can bring in all these other pieces associated with that specimen. What the habitat is like, where it was collected, what other species are in that habitat where it was collected, what its tissue content is, what its DNA is, and all kinds of information about where and when that specimen was collected. It can all be connected digitally. Imaging is another big task associated with collections. I have examples right now of 2D images and cleared and stained specimens here. Clearing and staining is really popular with ichthyology collections because the way that fish bodies are made, they respond really well to the dyes that are used. So that's something that really makes their anatomy stand out. 2D images are really important because they provide a digital image of what that specimen looks like, which people can use to verify identifications, and they can also use them in all kinds of training models as well. 3D imaging has really come into vogue over the last decade or so, and there's a couple of different ways you can do that. You can use an array of digital cameras to create a photogrammetric model of a specimen, or you can use a handheld laser scanner. With animals, we tend to use photogrammetry to accomplish 3D model building. Laser scanning tends to be more used with cultural objects like sculptures and pottery. CT scanning is like so hot right now. This is what the people are doing, and this is what we're talking about in collections. There's this massive project that just finished up based at the Florida Museum of Natural History called OVERT. And what they did was tested CT scanning capacity for many different kinds of vertebrates. CT scanning is only suitable for wet specimens because what you have to do is take the preserved wet specimen, dunk it in a special dye for a long time, do the CT scan, and then somehow they work this magic to leach all of the dye back out of these wet specimens. It's really incredible and cool technology. And the kind of information you can get out of that, that's my phone. I want to stop that. The kinds of information you can get out of a CT scan, just from the example here, I mean, bones, tissues, neurology, you can see their brains and their nervous system. It's really opening up a lot of possibilities for the use of these wet preserved specimens. There's always taxonomic support. Taxonomy is kind of a sister discipline to natural history. You collect more of these organisms, you want to describe them and put them in some sort of classification system, right? I've mentioned how fish taxonomy in particular is always changing. Earlier this year, there was a revised classification of ray finned fishes that built on classifications done in 2002, 2017, and 2021. This is constantly ongoing, and changing, and collections are a really important resource for this, particularly the genetic information that's associated with these specimens. What DNA has done for the ability to classify organisms cannot be understated. The information that we're able to get from genetic tissue collections is really vast. One use case that I really like with natural history collections, and this will come up a couple more times because I'm really interested in it, is tracking species populations and communities over time. A series of papers led by Prasanta Chakrabarty, the curator of fishes at the LSU Museum, in 2015, 2020, and 2024, he looked at the fish data that had been shared on GBIF in the area where the Deepwater Horizon oil spill had occurred, and he observed how it changed over that time. The figure that I put here, I didn't do this intentionally, but I caught it after the fact. The species on the top left there, Alosa alabama, that's the Alabama shad. That fish has disappeared from so much of its range now that it was petitioned this spring to be listed as an endangered species under the Endangered Species Act. Okay, so I've talked about our collections, and I've talked about what can be done with collections generally. What does that mean for us here at LUMCON? My primary directive right now is to finish our cataloging project. Our previous technician assigned catalog numbers to every single one of our specimens, and it's my job now to come in and get these numbers physically associated with the specimens themselves. With wet collections, the best practice is to put 2 labels of that catalog number inside the jar. We're also interested in pursuing QR code labeling on the lids of our jars to provide an interactive capacity with our collection. I'm obviously in a really rough draft phase of this project right now. Those finished labels will not be ripped pieces of masking tape. But this is something that will make the collection much easier to use and navigate when it's completed. Digitization is another important one. Again, I'm really grateful that I inherited a collection with such a robust digital record, but we want to complete the digitization process by getting that information onto the internet into some of these repositories where other people can use it. We're also working on transferring our data from single sheets in Excel to a relational database model in this specified software. Imagery is another big task for us. I did inherit this beautiful digital record, but I don't have a single image of anything in the collection, and there's 1,500 jars that we have to work with. Ideally, the way that would go is we would end up with an image of a specimen from every single one of those jars. That is quite literally years of work, and I'm happy to do that, but it will take quite a long time. The image on the top over here is of a squeeze tank. This is how 2D imagery is taken of fishes. The image on the bottom shows a photogrammetry setup for a wet specimen there. I'd really like to try 3D imaging of some of our specimens. With wet collections, you have to get kind of creative with photogrammetry. This is a fish that is hung from a hook from a turntable that's turned upside down. And then that turntable is spun, and those cameras take hundreds of pictures that a computer program then puts into a digital model. We can also use our collection to support taxonomy. One of the things we can do is provide the original and current taxonomic identifications with our records. This is something that has been lost over the years, and so we're working on rectifying that as we work on digitizing our data. A big part of my job will be keeping this taxonomy current. I will need to be checking the World Register of Marine Species database, or WORMS, pretty regularly to make sure that the names I have for the species in the collection are actually the currently accepted names. Again, this idea of assessing the history of a changing planet, we live in a really dynamic place, right? We've experienced a lot of change in our surroundings over the last 4 decades during the time that our specimens have been collected. If we need to go back to where we've sampled and see what those areas look like now and see what organisms might be there as a result of this change, how different is it or how similar is it? Using our collections as an outreach and educational tool. I really think the use of the collection to describe and explain taxonomy and the diversity of life is a really incredible opportunity for us. We've just moved into this building, right? If you check out our collections room tonight, it's looking pretty bare up front, but we do have some signage in the works, and we want to create a more visually engaging and presentable space for people to come visit. We want to build an interactive web portal so that people can interact with the digital data associated with our specimens, along with the specimens themselves. One of the things that our educators like to do with visiting students and groups is go collecting. It'd be really interesting to tag along on those trips and see if they collect anything new or interesting or exciting, and put some of that stuff into our collection, right? That's a neat opportunity. I also think that natural history collections have a really cool role in art. The art of natural history illustration, I mean, that actually is like kind of a lost art, right? It used to be everywhere, but digital imagery has largely taken its place. We could provide opportunities for that with the specimens we have. And then, of course, with the digital things that we're doing with our 2D and 3D imaging, that also supports artistic endeavors. We want to continue to grow the collection, right? I've mentioned the Oyster Reef Biodiversity Collection. We're also lucky enough to be inheriting the freshwater sponge collection of Dr. Mike Poirier. These are hundreds of specimens of freshwater sponges collected in the state of Louisiana and beyond. And they could really be an important resource for sponge biologists and sponge taxonomists. So that'll be really neat to have that here. Some future efforts that we want to do to help grow our collection include bioblitzes, just large one-day sampling events to see what's here. We can do those over time. And again, then we can start to assess how the area around LUMCON is changing. And we can do that survey work both here up in Homa and down in Cocodrie around our Marine Center. And then again, this idea of revisiting our historically sampled sites. So we have lots on the horizon for us, lots to do. It's a really exciting time for the natural history collections here at LUMCON. That's all I have for tonight. I'm happy to take any questions.
Any questions from our live audience? Or in person? I guess everybody's alive.
Except the specimens in the natural history collection.
Yes.
Yeah, so I have a question. So you said that to do the 3D imaging of the fish, like the wet specimen fish, you have to like hang it up.
Yeah.
Is that really like particular? Like, do you have to like put it back in the ethanol after so much time, like to prevent it to dry out?
Yeah,
Like,
so
how does that process look like?
there's a spray bottle involved. It's like a photo shoot for like a model, you know, you have to like spray the specimen and make sure that it's wet and it stays wet and it stays preserved, because yeah, those, if they're out of ethanol for too long, they can get pretty gnarly pretty quick. So there's definitely going to be some practice rounds of that for me, at least, to make sure that, you know, I can get a workflow going so that I can shoot the specimen pretty quickly and get it back in its preservative, because that is something to consider for sure. Yeah.
Any other questions? All right, we did have some online questions roll in during your talk.
Great.
So I will convey those questions to you from our online audience. The first one comes from Mary, who is interested to know how long the process is to actually preserve a specimen.
To preserve, it happens really fast. So when you collect something and you're out on a boat, the specimen comes off the boat, it gets euthanized. There are various ways to do that, all as absolutely humane and respectful as possible. And then that goes right into a fixative. These specimens are fixed in formalin. 10% formalin is the solution that is used. And fixing just means it preserves the tissues. And that's what takes the longest, I guess. They usually recommend that wet specimens sit in formalin for at least 24 hours if they're pretty small. And some of them can go, I mean, some of them can go up to weeks if they're big enough. But for things that we have in our collection, they'd be fixed in formalin for probably 3 days. And then they go in a jar of ethanol, and that's it. They're ready for perpetuity.
Perfect. Our next one is from Pete. Do you have to ever go outside of your in-house expertise to verify a specimen's ID?
Every day. Every day. Perhaps this is the part where I freely confess that my biological background is not in marine animals, right? So I'm learning all of this stuff right now. And it's fascinating. The resources out there on marine taxonomy are very rich and abundant, and they're approachable, right? I really like the worms database, marinespecies.org. They tell you a lot about, you know, the taxonomic tree and where things fit together and the history of the names of each of these specimens. Yeah, I'm always going outside for knowledge on taxonomy, and there's so much to learn.
The next one comes from Gavin. Really interesting question about the collection as a whole, but Gavin is asking if we have any LUMCON, the faculty, have any goals for the number of specimens that we want to reach in our collection.
The number of specimens.
And I'm just gonna Gavin, I apologize if I'm getting off of your question here, but maybe think about it as a diversity question,
Right.
like how diverse do you want the
Right, so
collection?
I'm thinking about, I mean, these aren't animals, right? So you don't want to just like collect to increase, it's not a numbers game, right? But it is interesting we want to be as taxonomically rich as possible, and we want to collect information and organisms from as many places locally as we can. And I say that it's a local collection, and a lot of local collecting has been done, but a lot of times those classes were going to the same few places, and there are other places around here where we've never been, and it'd be really great to go. So definitely focusing on taxonomic diversity and geographic variation to increase the size of our collection.
Olivia wants to know if you have a favorite specimen and if you've named it.
I have not named a single specimen. None of them have names. I did actually highlight my favorite specimens that are in the collection. I like the squid, and I also really like those red heart urchins. I don't know what it is about them. They're like balls of sand dollar delight. I just think they're so cool. They're so cool looking. I didn't know that that was a thing until I got into that collections room and found that junk. What is that? We have some dry ones of those too. They're pretty common, but I like the red heart urchin most of all.
Perfect. I'm gonna use that red heart urchin delight. Okay. Hope. I'm assuming Hope is a teacher, because Hope is asking if anyone can come see the collection, when they might be able to do that, and if we put our specimens out on loan.
I am here from 8:30 to 4, Monday through Friday. We are an open facility. I love visitors. Come by anytime. You could probably distribute my contact information if you'd like. Yeah, I'm happy to put that out there for anybody who's interested in scheduling a visit. And we'll totally do loans. Yeah, the logistics of that will have to be worked out, but they will be worked out and they will happen, because loaning specimens is a huge part of collections management. It's really important. Happy to do that.
Perfect. And Tiffany has a comment and then a question. Tiffany is very excited that there is something for her daughter to ultimately do career-wise, since she loves bones. So she's excited that there's actually a career field out there. But she's also asking how long those specimens last and if you have to maintain those.
Yeah, so the oldest one that we have in our collection is from 1966. Wet specimens, if preserved well, can last for well over a century, but they do require maintenance. Refilling and topping off preservative in those jars is a big part of my daily responsibilities. And periodically, especially if left untended for a long time, like many of our jars were, sometimes those jars are going to have to be replaced and the preservatives will have to be replaced. So there is a fair amount of maintenance that goes into preserving those specimens, but they can last for decades, many, many decades if they're preserved right.
Perfect. Any other questions from our in-person audience?
Yes.
I just have a question about collecting, specifically here in the Gulf of Mexico. How does it ever cross your mind of how concerned you are with time for collecting in the Gulf of Mexico, with the constant changes due to hypoxia and different things that are going on in the Gulf of Mexico, with the ability to still be able to collect out there? Does time concern you at all for this area?
Yeah, I think so. I mean, everything about how it's changing around here, right? I mean, it's a place that we need to get records of as detailed as we possibly can manage. And so, people someday can see what was here, in case what is here ultimately is not here anymore, right? Mm-hmm. Yeah, absolutely. Time concerns me. That's a great question.
Any other questions? Perfect. First, I want to thank our in-person audience for coming out to BlueWorks. Again, for those of you who are at home, the address for our BlueWorks facility is 335 Dixon Road in Houma. We will be doing future science talks, so we hope that people who are here in person tonight and people who are at home join us for those. We will be posting information about future science talks to all our socials as well as our website.
Thank you so much.
Thank you.
Did I do you justice?